Hey, good morning. Today we're going to be talking about proteins and DNA of the chromosome. This is going to focus on chromosomes, not gene expression. But we're going to be talking about a few things that we're going to go through kind of quickly that you've heard about before, but it's good to remember in terms of getting going with the rest of the class. So don't get too panicky if we talk a little bit quickly about certain things. We will be coming back to those. So it's kind of going to be a survey of a lot of things. There's a lot of words, a lot of just terms that you're going to have to just kind of learn. Okay, good. Okay, chromosomes are made mostly of DNA and proteins. Okay, so we talked already about each chromatid contains a single long double-stranded DNA molecule. We haven't talked at all about the proteins. Now, the proteins are actually what give the chromosome its physical shape and structure and organize it in such a way that it can be replicated and segregated during mitosis. So the proteins are very important. We're only going to talk right now about a few of these. We know that, of course, the protein, or the DNA rather, is wrapped in the histone proteins, and that's true all the time. But we're also going to mention cohesins, which are proteins that keep the sister chromatids together, and condensins, which are responsible for condensing the chromosome. This is a very active area of research, and there's not a lot known about most of these things. Keep in mind that these are not the only proteins that are bound to DNA ever, because DNA polymerase obviously binds there. You also have RNA polymerase, DNA repair enzymes, etc. So today we're just going to focus on the proteins that are responsible for what you actually see, which is that condensed physical X-shaped structure that's there in the microscope. Oops. Okay, I'm going to call this part 1, proteins of the chromosome. We're going to remember, first of all, the DNA is physically wrapped around a little bundle of histone proteins to form nucleosomes, and we'll talk about that a little more. But the DNA itself is not responsible for the shape of the chromosome. It's the proteins that are responsible for that are called the scaffold proteins, including cohesins, condensins, and other proteins that we don't really know a lot about. Okay, so the scaffold is what's left after the histone proteins, nucleosomes, have been removed. The DNA also remains attached to the scaffold in long loops. And this organization is kind of amazing when you consider what has to happen when a chromosome condenses and then segregates. Okay, cohesins are responsible for holding the two sister chromatids together. They are laid down during DNA replication and remain there holding the two together by means of a little circle until the time comes for their removal for segregation during mitosis and meiosis. Condensins are physically very similar to cohesins, except that they are involved in chromosome condensation rather than sister chromatid cohesion. This diagram shows cohesin addition during DNA replication. We remember that we start off in the G1 phase of a cell cycle, where each chromatid contains a single DNA molecule. During the S phase, replication occurs, so that now instead of having one chromatid attached to a centromere, we now have two double-stranded DNA molecules. These are called sister chromatids. Those are actually physically held together by little circles of cohesin that surround them that are laid down during replication, and they remain there until they are removed at the time of mitosis. Okay, and this is a very diagrammatic picture here. Early in replication, when replication starts, the cohesins are laid down along the sister chromatids and hold them together. Later in the cell cycle, when it's time to condense the chromosomes, remember that DNA replication occurs in S phase, and the chromosomes are not visible easily at that point. The chromosomes need to condense in terms of getting into mitosis, and they later will segregate. So it's the condensins that are responsible for this, condensing of the chromosomes for that process. Okay, so visualizing this is an experimentally purified chromosome, and it's been treated in such a way that we don't need to go into, that the nucleosome particles have popped off the chromosome so that the scaffold itself remains, but the DNA is organized into long loops. So the little X-shaped structure in the center is a scaffold, and the huge halo surrounding it is actually the DNA, which is still attached to the scaffold and forming these enormously long loops with the histones still gone. So we can basically infer that this sort of X-shaped structure is the scaffold proteins are responsible for that. But the histone proteins are responsible for condensing the DNA into the loops that are able to be formed. Okay, so what would the purified chromosome look like if we treated them with DNase before looking at them in the electron microscope? Reminder, DNase breaks the phosphodiester bonds in DNA. Okay, so if we did that, the DNA would be digested away by the DNase and will probably be released from the scaffold because it's going to be in little pieces. But the scaffold itself will remain because it's made of protein, not DNA. It will still look like a chromosome, but it will look like a very skinny chromosome. Okay, to be continued, proteins of the chromosome. We're going to move on a little bit and talk about nucleosomes and what those are doing. Okay, you've seen this picture before. It's a picture of little particles of protein where the DNA is actually wrapped around it to make this highly organized nucleosome core particle that itself can condense to form higher order structures that are the basis of what chromatin normally looks like. Okay, so histones are small basic proteins, and I'll get to in a second what I mean by that. Okay, there are 5 major histone proteins called H1, H2A, H2B, H3, and H4. Of these, H2A, H2B, H3, and H4 form the core octamer, and that's the part that the little octamer protein core is what the DNA is wrapped around. So this is the fundamental unit of chromatin. Okay, so I'd like to remind you something about proteins. This should be familiar, but it's been a little while since cell and molecular for some people, so I don't want to go through the basic stuff before we get too complicated. Okay, so of course proteins are made of amino acids, and the amino acids are linked together by peptide bonds. There are 20 common amino acids that are specified by the genetic code, although these can be modified post-translationally, as we'll see in a few minutes. And the difference among amino acids is in their R groups or side chains. And the properties of these R groups, they're different from each other in terms of their charge and their size. And of course, the proteins themselves are formed by the process of translation, whereby successive amino acids are linked together by the rules of the genetic code. Okay, so here's an example of histone H2B, which is a small basic protein. It's 126 amino acids long, you can see that, and it has a lot of the basic amino acids, lysine and arginine. Those are in red, and so you can see it's quite highly enriched for those basic so-called amino acids. All right, so this is written simply with the amino acids as letters. It's written in the sequence where the first amino acid, number 1, is the N-terminus of the protein, the first amino acid in the protein, extended to the C-terminus, which is the last amino acid protein. Amino acids are usually written by the single letter code for convenience, but not always. Okay, one thing we have to think about in genetics is that we're considering the amino acid sequence, and we don't necessarily need to know about the associated chemistry of the peptide bond. So very often, protein sequences are written simply as amino acid sequence in genetics. Okay, so the protein H2B is basic because it has a lot of lysines and arginines. As a result, it has a plus charge at cellular pH. The other histones are similar. So let's take. So we can see this amino acid shown. The side chains are shown in red on the bottom of the slide, and the core part of the amino acid, which is common to all of them, are in blue. So we can see the plus charges on these amino acids in both cases, and therefore they do have a plus charge overall at cellular pH. Now the rest of the amino acid, you wouldn't have a free carboxyl group or a free amino group because they would be linked together to form the peptide bonds. So we're just going to look at the side chains as far as this is concerned. Okay, so coming back to that, we've got our core octamer, 8 histone proteins, 2 each of histones H2A, H2B, H3, and H4. So on average, there's 200 base pairs of DNA per nucleosome across the genome. Although that does vary widely, there are nucleosome-free regions that occur sometimes. But what's actually physically interacting with the DNA is 147 base pairs of DNA is wrapped around the core. And if there is an H1 present, it will be outside the core, and it's called the linker histone for that reason. Okay, the nucleosomes themselves form the 10 nanometer fiber, which condenses or coils up further to form the 30 nanometer fiber, and that 30 nanometer fiber coils up even more to form the thicker fibers that you see in mitotic cells. And not much is known about those thicker fibers, really. Okay, so here's a sort of diagrammatic picture of nucleosome organization. The top shows the nucleosomes in their form of their 10 nanometer particle, where you can see this little cylinder, that's the histone core, and the DNA is wrapped around it, 147 approximately base pairs per core. You've got a nucleosome-free region to indicate that there's some regions, especially in promoter and regulatory regions, where the nucleosomes have been shifted aside. And then you've got the higher order structures where the nucleosomes have been organized into higher, more complex configurations. Okay, notice also the little lollipop things on the tops and bottoms of these little cylinders. That indicates the fact that some of your amino acids within your histone proteins are modified in various ways, which are very actually important for things like gene expression and also for chromosome condensation. Right, so the most typical modification that you'll hear about a lot is acetylation. So a regular lysine has its plus charged amino acid. Notice this amino acid is turned upside down compared to the other one, but here's your plus charge. That can be acetylated, which is say an acetyl group is added to that nitrogen, and that obliterates the plus charge that would have been present. So acetylation by histone acetylases catalyzes that reaction, but it's a reversible reaction because the acetyl groups can be removed. Okay, so these nucleosomes here, this is a view of from the top of a nucleosome where the DNA encircles the histone proteins and the core is written underneath. So the little dots indicate the fact that the amino termini of these histone proteins are where most of the modifications are found. So if you're going to acetylate your histone, it's going to be on lysines that are largely in the first, essentially, part of the histone protein. Okay, here's a clicker question. The answers to these are in the last slides at the end, by the way. Nucleosome core particles contain blank different types of histone proteins. Okay, so the answer is D, 4, right? The nucleosome core particle contains blank total proteins, including blank molecules of histone H3. Okay, it contains 8 total proteins, 2 each of H2A, H2B, H3, and H4. So the answer is D. Modifications of histone amino acids are most commonly found near the N terminus of the histone protein, in the middle of the histone protein, near the C terminus of the histone protein, throughout the length of the histone protein. Okay, so the answer is A, right? Okay, so this is part 2, which I'm calling DNA of the chromosome. Okay, it's basically the point of this is to get a feeling for what our genome is like in terms of the types of sequences that it contains and something about what their functions are. Okay, we think about protein coding genes as the real thing, which is to say those are the DNA sequences which eventually give rise to the formation of a protein, which catalyzes a reaction in the cell or serves a structural component of a cell or something of that sort. But it turns out that protein coding genes are only a fraction of the DNA that we have, and there's a lot of other things that we need to know something about. Okay, we got lots and lots of repeated sequences in our genome, including transposons and pieces of them. And there are a lot of simple sequences that do not actually encode proteins, but can serve other functions. And I'm going to go through a few of these in a second. And finally, there's RNA encoding genes. Now, of course, we've got plenty of RNAs in our cells, tRNAs, RNAs, etc. Now, those are, of course, encoded by the DNA, but those RNAs are not translated. They don't produce protein. Their job is to function as RNA. Okay, so this is a quick trip through what's a protein coding gene. Okay, it occupies a particular location on a particular chromosome. We talked about maps. It includes a promoter, regulatory regions, in addition to the part of the DNA that's actually copied by RNA polymerase by the process of transcription. We know that a pre-mRNA will be formed by copying the template DNA, and that itself is processed because it's got introns in it that need to be removed before we can put the exons together, which is, of course, where the actual translated codons are located. The mature mRNA is then exported to the cytosol for translation according to the rules of the genetic code, and then the protein performs its function in the cell. So a protein coding gene is what we really think of as a real gene, but there are plenty of other things that are real genes also, and there are also a lot of genes or DNA sequences which don't encode anything specifically, but still have very important functions in the cell. Okay, so here's a not necessarily great representation of what genes are like. The point that's being made here is that in bacteria, for example, genes are packed very tightly together, and when you start one gene, the next one stops. They're very, very close. In yeast, genes are slightly larger, and there's a little more space between them, but not very much. But as you go up to human, you notice there's, first of all, a lot of space between genes, and that there's little green areas here which indicate what is protein coding versus what's intron. So we gain introns as we get to be human, but we also end up separating our protein coding regions into lots and lots of little pieces. Okay, so what's not true about this slide is it makes it look like human genes are equally spaced with some similarity between the genes and spacing. Well, that's not true. You can have some regions of the genome which is closely packed with genes, which are right next to each other, or else you can have large regions where there are no actual protein coding genes at all. So it's a good slide to represent the fact that introns are very prevalent and maybe even more than the exons in terms of sequence, but it's not so good in representing what the genome is like on a whole, on a larger scale. Okay, in the previous picture, the little green stripes were the exons, and the white part were the introns. The white parts between the genes are intergenic, and these can do very important things, but creating protein is not one of them. Okay, so for a scale of miles here, genes do vary a lot in size and intron number. The top gene is a human beta-globin gene, and it's only 2,000 nucleotides itself, and has only 2 introns. And the human factor VIII gene, by contrast, is more than 200,000 nucleotides long, has 26 exons, and most of its sequence is actually intron, right? So the orange part, the large part, those are the introns that are going to get removed. And when you put the exons together, you've actually discarded most of the original transcribed region of the gene. Seems wasteful, but that seems to be how we do it. Introns are much more prevalent than exons, in fact. About 24% of our DNA is actually intron. So even though it's within a protein coding gene, it does not itself encode protein. And so that leaves us with about 75% of genes, or DNA sequence rather, that's not in a protein coding gene at all. So intragenic is only about 25%, meaning within genes. Outside protein coding genes is 75%. So there's an awful lot of stuff out there, some of which is very significant, and some of it is we're still trying to figure out what exactly it's doing, if anything. Okay, so the rest of our DNA that's not in exons, clearly we've got introns. They don't encode proteins, but they can have important sequences within them. We also have DNA sequences in the spaces which do serve other functions. Okay, a lot of repeated sequences occur, some of which are very important, namely those involved in chromosome maintenance, and we'll give the examples of telomeres and centromeres in a second. And there's a lot of repeat sequences which can be very useful to us for DNA testing purposes, for example, but have no real function on their own. But we certainly can't forget the RNA encoding genes, because those are not translated, but they certainly produce RNAs that are incredibly important in the cell. We also have lots of transposons and relics of them, and also relics of decaying genes, which are called pseudogenes. Okay, heterochromatin versus euchromatin. Heterochromatin consists of tightly coiled chromatin that does not loosen during the cell cycle, right? Euchromatin is a usual kind of chromatin that changes its condensation state during mitosis. Okay, heterochromatin is always darkly staining, implying that it's tightly coiled. But that doesn't mean it has no function. The alpha satellite is located at the centromeres of all human chromosomes, and it's a heterochromatic appearing repeat of 171 base pairs that repeats over and over and over in tandem at that location. The telomere repeat is similar. It occurs on the extreme ends of chromosomes. We saw a little bit about telomeres last time. And it, of course, itself functions importantly in chromosome replication, and it's required for maintaining the length of chromosomes at its ends. So the important point about this is these are repeated sequences, heterochromatic, but they don't encode proteins. But certainly maintaining your chromosomes is an important function that we need to be able to do. Okay, so the alpha satellites, 171 base pairs, repeated over and over and over, and it's responsible for binding proteins to form the kinetochore, which is of course what binds the spindle so that the chromosome can segregate during mitosis and meiosis. So here is a picture which represents the 171 base pair alpha satellite repeat at centromeres, and a representation of what it's doing there, which is to say binding proteins that will contribute to the ability of those two chromosomes to bind the spindle and separate from each other when mitosis occurs. So this is clearly an important function in the cell. Telomeric DNA is also an example where a repeated sequence serves an important function. In this case, it's replication of the extreme ends of the chromosomes. The telomeric repeat in human, as we saw last time, is sequence 5'TAGGG, and it goes on for several hundred, sometimes thousands of base pairs at the ends of chromosomes. Presenting that, we saw this picture before. The little yellow dots are the telomeres because we saw them by fish, and the blue part is the rest of the chromosome. So these repeated sequences are found at the ends of our chromosomes. And they don't encode protein, but they are useful, right, because they actually are responsible for replicating the ends of chromosomes. It does have an extremely interesting structure at the very end where there's a single-stranded tail which actually inserts itself into an adjacent piece of DNA, forming this T or D loop type structure, which is unique to the telomeres. Okay, remember the ends of the chromosomes are replicated by telomerase, which is a specialized RNA protein complex, which is required for replication of this region specifically. Okay, so besides the sequence repeats like telomeres and centromeres, which makes sense, where they actually serve a very clear function, there are the dispersed repeat sequences, which are short repeats that are everywhere, just everywhere in our genome, not just in a heterochromatic region. These are sometimes called microsatellites, and they're sometimes called STR for short tandem repeat loci. And these do occur in variable from one person to another, which is why they're useful for DNA testing. And we'll talk a lot more about that when we do DNA testing, etc. Okay, transposons are amazing. They're a humongous part of the proportion of our genome. These are mobile genetic elements, and they have no fixed locations, and they can move to new ones, unlike regular genes, even STR loci, which are basically fixed and have typical map locations. The transposons that we have do not transpose very frequently, fortunately, but they do, and when they do, they can cause chromosome rearrangements and sometimes genetic disease. Okay, the types of transposons we have are very many. These are the signs, short interspersed nuclear elements, meaning 300 base pairs or less. We have the lines, which is to say long interspersed nuclear elements, and those are interesting too. We also have the virus-like retrotransposons and a few DNA transposons. And these are also pretty much everywhere in our genome. Okay, the most well-studied human transposons are a line called L1 and the ALU example of the signs. Now these are very important in the evolution of our genome because they can rearrange genes and chromosomes, and they do sometimes cause disease. But mostly they don't transpose, fortunately. Okay, so here's a representation of a gene. I forget which one it is, but it's got 14 exons, which are shown. The light green is the introns. Like many genes, the introns are very huge compared to the exons. So after transcription, we have to splice out all those introns to get all those 14 exons together. But my point at this moment is that our introns are just peppered with transposable elements and pieces of them. The ALU, the first line, are the example of a sign, this short interspersed repeat element. And this particular gene has 1, 2, 3, etc. All these within its introns. Other signs, bits of lines, and various other bits of things are within our introns. They don't cause any harm usually. They certainly don't change the protein coding sequence, but they're there just kind of hanging. Okay. So there's an awful lot of transposable elements in our introns, and sometimes you can attribute certain chromosome rearrangements that have happened to rearrangements between the transposable elements rather than the actual DNA of our own genome. Okay, here's a clicker. The alpha satellite sequence occurs, dispersed at random locations throughout the genome, at centromeres of all human chromosomes, at telomeres of all human chromosomes, within introns of many protein-coding genes. Okay, so the answer is B, right? It's a centromeric sequence. The proportion of the human genomic DNA that does not encode protein is closest to 99%, 90%, 9%, 0.9%, 0.09%. Okay, so the answer is A, right? 99%. Okay, lastly, we have my favorite, genes that encode RNAs that are not translated. They're not going to go to the ribosome and produce protein, but they're still extremely important. Probably more important than protein. Well, not more important than protein-coding genes, but very important. Okay, clearly we've got the tRNAs that are responsible for translation, the RNA component of the ribosome, which actually contains its catalytic activity, the uRNAs that form components of the spliceosome. We've got SNO RNAs, which are for modifying RNAs. We've got many, many regulatory RNAs of different sizes and types. The microRNAs for regulation of translation and RNA degradation, and then several long non-coding RNAs, which can be responsible for forming heterochromatin and other aspects of gene regulation. So here's a typical tRNA. It's encoded by the genome but doesn't get translated. Its job is to function as a tRNA. Remember, there's the anticodon loop, which is going to bind the codon, and we've got the 3' end where we're going to bind our amino acid for participation in protein synthesis. But this itself is not going to produce protein directly. Similarly, we've got the ribosomal RNA genes. Now, of course, we've got ribosomes, and they're made of proteins and RNA. And this just shows you the incredibly complicated secondary structure where the ribosomal RNA base pairs to itself to form this enormously complex structure. Okay, it's so important, it's even got its own RNA polymerase. Okay, regulatory RNAs include EXIST, which is a long non-coding RNA for transcriptional regulation, and also many, many microRNAs for translation or RNA degradation. Okay, EXIST is an amazing RNA. It's very well studied, and even more things are being found out about it now. But it's produced from a region of the X chromosome, and it's an RNA, and its job is to coat the inactive chromosome, the inactive X chromosome, in mammalian females. Okay, so mammalian females have 2 X chromosomes, but only one is active in any given cell. And the process whereby one of the X's is inactivated involves this EXIST non-coding RNA, which binds to one of the chromosomes, one of the X chromosomes, as part of the inactivation process, and leads to the formation of heterochromatin, so that particular X chromosome is not active in any cell that derives from that particular cell. Okay. MicroRNAs are regulatory RNAs. They are small, only about 21 to 20 nucleotides long in their mature forms, and again, they are not translated. But they do very important things, like determine whether a particular mRNA is degraded or if it's translated, and they're very important for certain developmental processes. Okay, so for example, if the ones that are encoded by the genome form little teeny RNAs that can either bind mRNA, particular ones that are complementary to itself, and cause degradation of the RNA. They can also bind to the mRNA and cause its translation to be inhibited. And they also contribute to silencing of some genes. So even though they're small, they don't produce protein, they have huge effects on our genome and how it's regulated. Okay, so the remaining slides have the answers to the clicker questions that we just had. Okay, see you next time.